members. Okay, so thank you for the opportunity to speak here today. Obviously a short time to present a lot of science, a lot of development, a lot of manufacturing development and business as well, but I'll try to take you through our story. If I can get my slides to advance, that's better. We're a public company on the Paris Stock Exchange, IPO'd in April this year, sorry, so there's our disclaimer. And with respect to Helen, there is a third arm to the development of T-cell immunotherapies, and we believe, at the forefront of that one, which is using regulatory T-cell immunotherapies to treat chronic refractory autoimmune diseases, utilizing their natural activity, and really focusing on making them as well, while personalized autologous being economically viable. So in terms of our strategy and positioning, we see ourselves as having an economically viable, personalized T-cell-based immunotherapy platform, which can create a number of different products. We have our own GMP-certified manufacturing facility in France, and we have a very strong IP base with over 140 granted patents. We're already advanced in the clinic in Crohn's disease, our first indication, where we're looking at the 100,000 patients in the 7 major markets each year who are refractory to all existing therapies, and I'll show you some of the clinical results there. And this original business model that we have, which is based, as I will show you, on the fact that we can manufacture one time to create multiple years of treatment for these patients with these chronic autoimmune diseases. Our first program is validated by a partnership with Ferring Pharmaceuticals in IBD. And building off that, we are now taking forward programs ourselves in orphan diseases, which we will retain to build the value for the company, the first of those being autoimmune uveitis. So this is our platform and our pipeline as it stands today. Our first product in mid-phase 2 for Crohn's disease, and very shortly to go into a phase 2b trial. Our second product, the Ag-Treg, coming into the clinic in uveitis in the first half of next year, and behind it a platform of other products. What distinguishes the products for us is we make our Treg products antigen-specific. And so the first part of each product name that you see there, ova-Treg, col-Treg, myelin-Treg, etc., refers to the antigen specificity of the product. And the key point of that is we use that antigen specificity. We choose an antigen that is co-localized with the inflammation of the disease to activate the activity of our Treg cells at the sites of inflammation for that disease. So that creates disease site specificity, and product specificity. So how do our autologous antigen-specific Treg cells work for inflammation and autoimmunity? The first thing that happens, if I can make this pointer work, great. The first thing that happens is the cells naturally migrate to the site of inflammation. That is the natural role in the human body of the Treg cell, and particularly of the type 1 Treg cells that we work with. So they're injected intravenously, they circulate, they work out where the inflammation is, and they migrate to there. Once they're there, they come into contact with, as Professor Schneck said very nicely earlier, the conductor of the immune system, the APCs, and here we get local antigen-specific activation of the cells. So they are presented to the APC in the presence of the antigen, which we know to be localized at the site of inflammation. So ovalbumin, for our product Oversave for Crohn's disease, ovalbumin, which you will find distributed in the digestive tract. They get activated and they proliferate as well, locally at the site. We then move to the third step, which is actually the inhibition of inflammation. And here again, as everybody knows who's at this conference today, using the power of cells is really very different from using single target molecules to treat diseases. So we have all the power of the Treg cells that they use naturally in the body normally to control our reaction to antigens, to inhibit inflammation through multiple cellular and molecular targets. We can talk about release of soluble factors like IL-10, IL-13, cell-to-cell contacts to inhibit the activity of pro-inflammatory players through molecules such as CTLA-4. We can talk about the downgrading of the inflammatory metabolic activity through degradation of ATP with CD39 interactions. And we can talk about direct cytotoxic activity on cells like the monocytes or the dendritic cells in the area of inflammation. So we are really unleashing a cascade of anti-inflammatory activity here. And then finally, and this is really very different as well to what I see as the flip side of the coin of T-cell immunotherapy, which is the CAR T-cells and the TCRs of Adaptimmune. These cells do not stay around. They do not create a memory. They are cleared after about 8 to 12 weeks from the body. This is very good from a safety viewpoint because you're not having the risk of tumorigenicity. It also implies that one would need to retreat chronically, and you will see that is in fact the case. So putting all those factors together, we believe we have everything that should lead to high efficacy, very good tolerability because these are autologous cells and there is no question of anything like cytokine release syndrome with these anti-inflammatory cells, and low resistance due to these multiple cellular and molecular targets. Again, big topic here today. How do you produce viably, economically, and in a way that works for the health system? We start actually from a very simple 150 ml blood sample, which is shipped to our manufacturing site where we separate the white blood cells. And then we have a process that we refer to really as an education of the cells. We do not do genetic engineering of the cells. We get them in the place, we put them in with an antigen, the chosen antigen for the product, ovalbumin for our first product, collagen type 2 for our uveitis product. In the presence of a cocktail of cytokines and growth factors, we then see antigen-specific cell enrichment, and from there, we then separate out those type 1 Treg antigen-specific cells that will form the drug product. Today, we do this by limiting dilution. As we go forward, we'll do this by direct selection, therefore speeding up our process and reducing its cost even further. Once we have those cells, those antigen-specific Tregs, we then move to an expansion phase, classically, and at the end we have a bulk product, which we freeze into individual doses, which form the actual final drug product. Our cells, as you will see from our data, are very, very potent. They're also very, very robust, and they can be stored frozen. We now have data going up to 7 years storing these cells, which means that from one manufacturing cycle, we can easily create enough drug product to treat a patient, for instance, for 5 years. And I think you can see in terms of an autologous immunocellular therapy, that completely changes the economics. We'll amortize that cost of production at the start to have a very good ROI over the period of treatment of the patient. And of course, having these individual frozen treatment doses, you will see, is going to lead as well to a very nice approach to actually getting the product to the patient, the doctors using it as well. Just to say, as I said before, it's a validated process, certificated and accredited by the French ANSM. We do, of course, continue to optimize it. And even as we're now about to start our phase 2B trial, we're already now looking at the next phases of how we are going to really outscale it and automate that process. And of course, we have high barriers to entry on this through our know-how and through our patents. Looking now at the first validation of our approach clinically in Crohn's disease with Oversave, our lead product. We're positioning ourselves in Crohn's disease, as we will, in fact, for really all indications we're looking at currently, at the end of the current treatment pathway, if there is one, of course. For some diseases, there may not even be a treatment pathway. So for Crohn's, we're looking at being a fourth-line treatment, if you like. Another way of putting it is a rescue therapy for those patients who fail all of the therapies. That is, patients who today have taken steroids, immunosuppressors, have failed on anti-TNFs and other types of biologics that are now coming to the market. Patients who have a very high annual cost of treatment and in terms of pharmaceuticals, but also generally in terms of care, and for whom we want to really bring a product that will have excellent efficacy, very good tolerability, and really add value to them and to the medical system. An important sized market on the basis of the current costing of anti-TNFs. The data from our phase 1/2 proof of concept trial, 20 patients with severe refractory Crohn's disease, including to anti-TNFs, treated with either 10 to the 6, 10 to the 7, 10 to the 8, or 10 to the 9 cells, each patient with 3 months follow-up. And indeed, later we went on to repeated injections in some patients after a protocol amendment due to the benefit seen, which you can see on the right-hand side of the graph. This is the data from our best dose, which actually turned out to be 10 to the 6. Our products have a bell-shaped dose response curve due to the interaction with the APCs that I told you about earlier. And you can see here in these patients who no longer respond to any treatment, we saw a 75% response rate at week 5 and week 8 after the single injection on the regulatory gold standard endpoint for Crohn's disease, which is a measurement of clinically significant improvement. And indeed, in these very ill patients, we even saw getting close to 40% at 5 weeks of patients who became asymptomatic on this treatment. So really a high response and a high remission rate. I won't go in detail over this slide, but just to show you, looking at the measurements along the time now, so the 12 weeks of follow-up, you see on the Crohn's disease activity, the primary endpoint, you see a quick drop in disease activity, maintained out to 8 weeks, and disease activity starting to come back, symptoms coming back at 12 weeks. Not a surprise, because this is when the cells are disappearing. Consistency of this data in quality of life on an objective biomarker, C-reactive protein, and on an immunological biomarker of cell response to antigen, which also will form the basis for us as a biomarker. And just to say very quickly, independently, KOL, with caveats, classing this data as remarkable. Our next study is going to be launched before the end of this year, phase 2b study in Crohn's disease, randomized placebo-controlled, 4 arms, 10 to the 4, 10 to the 6, 10 to the 7 versus placebo, 144 patients, 2 injections double-blind, 2 further injections open label, starting, as I said, very shortly with results in a couple of years' time, and that will really confirm the proof of concept and set the ground for the phase 3. A few words about uveitis, the next indication we're moving into. A leading cause of blindness in the developed world, an orphan disease where we will follow. We expect a typical orphan development process. Posterior autoimmune uveitis is poorly treated. Steroids are virtually the only approved product, with some single-target mAbs in development as well. It's a disease characterized by a dysfunction of the Treg compartment, and we've already shown efficacy of col-Treg in a model. The antigen chosen is type 2 collagen because it is present in the interior of the eye. So when the cells arrive, migrating to the inflammation, they will be activated in the presence of type 2 collagen and release that cascade of activity I spoke about before. It's manufactured using the same phase 2b process as Oversave. Therefore, we're benefiting from all that investment that's going on in there. It's an ATMP, and we're looking for orphan designations to be granted around the end of this year. And we will undertake a placebo-controlled multi-center trial starting first half next year with results expected a year later. Just quickly on our business model, if I can get the slides to change, which I can't. Interesting. But very quickly on the business model, you can see that we have, we'll be looking for a product to be available 5 weeks after prescription, frozen in individual doses, a biomarker for response after first injection based on that antigen response I referred to on the Oversave data. Looking at a cohort of patients, even if you're aggressive on attrition over time, manufacturing cost, revenue for most of those patients, not all, because those patients who are shown not to be responders, we can say we will not get revenue. After 3 years, even if you cut the model at 3 years, even though patients can be treated longer, you see that we have revenue versus cost of goods, and we're expecting a margin in the region of standard biologic products for our product, which will also be very easy to use for the physicians. We have a strong news flow coming up based on the uveitis program and based on the Oversave program, as you can see. We have a good cash position, 17.4 million of cash at the half year this year. And just to sum up, we really believe that we're developing personalized T-cell immunotherapy for autoimmune and inflammatory disease that's economically viable on an industrial scale based on the technology platform, validating off our first and building off our first indication already with a partnership to validate that first, an orphan focus for the company itself to build value, and this unique business model with the limited cost of goods and the strong margins. Thank you very much.